let us see the topic codominance. So codominance means together. So this pattern occurs due to simultaneous, that is joint expression of both alleles in the heterozygote. The phenomenon in which two alleles are both expressed in the heterozygote individual is known as a codominance. So in the F1 generation is a heterozygote nature, means the both the parental character are expressed together, that is codominance. Examples of the codominance. The first one is the red and white flowers of camellia. So camellia is a tea plant which has a two different flower character. So one is a flower producing character. So one is a red flower, another one is a white, means in the F1 heterozygote nature. So both the character is expressed, that is a double color flower is produced in the F1 generation. The another one is the inheritance of sickle cell hemoglobin. So here also in the F1 generation both the normal and sickle cell of the hemoglobin is present in the F1 generation. And next one is the ABO blood group system in human beings. So here also both the alleles of IA and IB is expressed in the codominant in the F1 generation. This is also expressed in the law of segregation of the Mendel's. In plants, the codominance was demonstrated with the help of the electrophoresis or chromatography for the protein or flavonoid substance. So I hope you already know about the chromatographic technique. So we already studied in the 11th standard also. So in the electrophoresis technique, this is the another one of the way to understood the codominance. In the electrophoresis technique, this is the agarose cell electrophoresis by the help of the current, which produces the different banding pattern of the plant. So that is the electrophoresis technique. So for here the example is a Gossypium hirsutum and Gossypium strutianum. So this is the two different other same genus and different species of the cotton plant. So in the F1 hybrid shows the presence of the both the type of protein similar to their parents. So both the parent have the different banding pattern for their seed protein. But in the F1 hybrid shows the both the parental character. So both the banding system. So by the help of the current in the electrophoresis techniques, it releases the both the seed protein of the parental character. That is the together characteristics will expressed in the F1 generation. So heterozygote nature. In the F2 generation, it produces the both phenotypic and genotypic ratios are remaining same. That is a 1:2:1 in the F2 generation. So what is the main difference between the incomplete dominance and codominance means? In the incomplete dominance, F1 generation is a intermediate color, that is a red flower and white flower. It produces the intermediate pink color is produced. But here the codominance, both the color, that is a double color flower is produced. So this one is the main difference between the incomplete dominance and codominance. But here the F2 generation, both the phenotypic and genotypic ratios are same, both in incomplete dominance and codominance.